Hello and welcome to the second part of Chapter 2. This is the lecture for my Biology 160 class, my being Christina Howard's class, but anybody else is welcome to use this information as well. So let's get started. But first, of course, the all-important does my pen work test. And let's do green today. Let's see. Hope for the best. Rad. All right, let's boogie. Okay, so this PowerPoint is really all about the different kinds of molecules that life is made of and that life relies upon to remain living. So getting started, let's talk about organic molecules. So organic molecules, in science, organic means made of carbon or made with carbon. It does not mean grown without pesticides. It does not mean someone saying to the plants to help them feel good enough to grow. It does mean carbon-containing molecules, and specifically hydrocarbons. So molecules with hydrogen bound to carbon. So the simplest hydrocarbon is methane. So remember, because of the valence needs of carbon, it likes to form 4 covalent bonds. So it has 4 electrons in its valence shell, and in order to get its sweet octet, it wants to have 8. So it's going to share a pair of electrons with each hydrogen it comes across until all of its bonds are met. And notice that because there is this axis of symmetry and this axis of symmetry, this is going to be a non-polar molecule. So there's going to be equal electron sharing all around. So let's just remind ourselves what carbon's valence shell looks like. And that's because, hey, you just learned about valence shells. You need to keep track of valence and electrons and protons, so it makes sense to add this layer on top of the stuff that we're doing that's a little bit more sophisticated. So carbon has an atomic number of 6. So at its nucleus, it has 6 p's and 6 neutrons. Now, neutrons don't contribute, but I'm drawing them just for posterity's sake. And then in its inner shell, it's going to have a 1 and a 2 electrons, and now that shell is full. And in the outer shell, it's going to have 1, 2, 3, and a 4. So the shared electrons are going to be electrons that hydrogen contributes. So if we were to draw this methane out, there's a hydrogen. There's one. There's one. And let's put one down here. Perfect. All done. Okay, so organic molecules are based on hydrocarbons. And here's the same picture as I just drew, but with a little bit less detail. Okay, so hydrocarbons can take on very complex forms. So they can go from everything from a fatty acid, so this happens to be stearic acid at the top here, to an amino acid, and sugars. So this happens to be glucose, a monosaccharide. So although hydrocarbons are relatively simple, so they primarily consist of C's, H's, and O's, which is a very small subset of all of the available elements, there's many, many ways to arrange these, and there's lots of ways you can play with the properties of these molecules depending on how bonds are arranged and where you put things. So for example, these two are acids because for each of them, this hydrogen and this hydrogen dissociates into solution to become a hydrogen cation. Glucose is a sugar that your body relies on heavily for fuel, and it is arranged into a 6-sided ring, so we call it a hexose sugar. So there's lots of variety, even though the ingredients for hydrocarbons are relatively simple. So I just showed you a fatty acid, which is a component of lipids. I showed you an amino acid, which is a component of proteins, and I showed you a carbohydrate, glucose. So these are 3 of the 4 biological macromolecules. So remember, A molecule is a compound that is held together by covalent bonds. And macro means big. So these can be very large. So how big is big, since we're thinking about things that we can't see with the naked eye? So what would make something that we can't see with the naked eye big? Well, compare them to gases like H2, which is just two hydrogens bound to each other. That's little. This is big. So at the top here we have the class, which is just the general designation. And then in this row we have the elements that make it up. Monomers are a building unit of polymers. So a polymer is the name for a complex molecule that has a repeating arrangement of smaller molecules. So monomers make polymers when they are bound together. And then we have function as well, and a visual representation, so picture. So for carbohydrates, the elements are necessarily and obligately, meaning they have to be in there, C, H, and O. And for carbohydrates in particular, the O's are numerous. And I'm going to make that distinction because there's some other cases that we'll get to in a minute. So monomers of carbohydrates are monosaccharides. So these are things like glucose and galactose. And the polymers are disaccharides, so two monosaccharides together, and polysaccharides, which is more than two. So these are for energy storage and also to contribute to cellular structure as well. So here is a glucose chain in a three-dimensional space-filling model. Lipids also have carbon, hydrogen, and oxygen, but compared with carbohydrates, there aren't very many oxygens. There are also occasionally phosphorus atoms and phosphate groups. Lipids aren't really monomers and polymers, and we'll get to that more later, but fatty acids are kind of like monomers, and you can add and subtract them to lipids to give them different properties. So we'll put a pin in that for now because it's something we'll come back to. So these are for long-term energy storage, so things like subcutaneous and visceral fat. We also use them to build cell membranes and to waterproof ourselves. So here's a molecule of cholesterol, which is not as evil as health officials make it seem. I'll explain that later, but that is what it looks like. Proteins have carbon, hydrogen, and oxygen, but they necessarily and obligately, meaning have to, contain nitrogen. So nitrogen is a defining element of proteins. The monomers here are amino acids. And you saw an amino acid in the previous slide. So amino acids, you might already be familiar with some of these because they tend to be present in supplements. So these are things like lysine or tryptophan, which is the amino acid in turkey that makes you feel sleepy after you eat it, or if you eat too much on Thanksgiving, which is coming up. Happy Gobble Gobble Day pretty soon. All right, so when amino acids get together, we call them polypeptides. So poly means many, and peptide actually doesn't refer to amino acids. It refers to the kind of bond. So I'll write kind, not kid. Bad Christina. Kind of bond. So a peptide bond is a particular kind of bond. You'll see those later. And so when amino acids bind to each other and there are more than one peptide bond present, we call them polypeptides. And these have lots of diverse functions. So they can be enzymes, like the enzymes that perform the biological reactions that keep us alive, or they can be simpler and more strand-like. So keratin is a protein that makes up our hair and fingernails. Nucleic acids necessarily contain all of these elements. Their monomers are nucleotides, and the primary ones that we're going to talk about for now are DNA and RNA. So these might be familiar to you. So DNA is where genetic information comes from. So we store and express the information contained in our genes by accessing DNA, which has all that information in it. And then RNA is made out of DNA, made from DNA. So we'll get to that later. And RNA is used to make proteins. So, how do you make a polymer from some monomers? Well, there are two reactions, and these are formalized reactions, meaning that these are general. So we're not saying that this reaction set is specific to carbs, to lipids, or to proteins or nucleic acids. This is just a generalized reaction form in biology by which polymers are synthesized and by which polymers are broken down. So notice that each of these monomers has a hydroxide group. And if you look at the hydroxide group, if you pull out two H's and an O, you get water. So if we dehydrate something, that means we're taking water out of it, right? So if I dehydrate these two monomers, so I pull this water out, now I'm left with the two monomers I initially had, and they're holding hands. So it's cute that the oxygen that they're linked by looks like a little stick figure hand holder. Okay, so that's how you build a polymer. The opposite case is when you break polymers down. So why would you want to do that? Well, you are doing hydrolysis all the time so that you can digest and absorb the food that you eat. So let's say I have two monomers, and let's say for the sake of argument that this particular short polymer is lactose. So lactose is glucose bound to galactose. So if I want to break glucose and galactose up, and the reason I want to do that is because this molecule right here, this disaccharide, is not absorbable by humans. So we don't have the correct transport mechanism to get this into our bloodstream. If we want to reap the benefits of this nutrient, we have to break galactose off of glucose, and then they can enter the blood separately. So you got to break them apart first. So hydrolysis, hydro means water, and lysis means to break. So this is a reaction where you use water to break apart two monomers. And this particular chemical reaction is facilitated by enzymes. And actually this one is too. So an enzyme is a protein that catalyzes reactions. So to catalyze means to facilitate or make easier and more likely to occur. Oops. I want to go back. There we go. Okay. So I want to break lactose up. I need to use an enzyme to do that, and the enzyme I'm going to use is lactase. So the reason I underlined ase is because this is the suffix that we use to denote proteins that are enzymes. And in this case, it's easy to tell what this enzyme does because it's lactase. So lactase breaks up lactose. Duh. If you're lactose intolerant and you decide you absolutely positively must have the cheese plate, you can actually take lactase in a pill form with your meal to help digest some of that lactose for you to mitigate or minimize your lactose intolerance symptoms. So lactase takes a water molecule and shoves it in this little space, and that results in the monomers going back to their separate forms. So this becomes just glucose by itself, and this becomes just galactose by itself. Okay, so now we can move to the next slide, and that is carbohydrates. So the next few, actually the rest of the PowerPoint really is going to get very nutritional in nature, because the reason that the food pyramid that the FDA makes is the way it is because all of the biological macromolecules are also molecules that we must eat in order to rebuild and fuel our bodies. So of the carbohydrates, and here we have a bunch of natural ones, so these are some sprouts, it looks like, and some grains. So monomers, or monosaccharides, these are simple sugars. So they are individual units. They don't need dehydration synthesis or hydrolysis. They're already by themselves and readily available and absorbable. your diet should include more complex carbohydrates because your body spends more energy digesting these, and they're a healthier source than pure sugar. So the reason that you shouldn't have too many simple sugars is because that can cause weight gain and diabetes. So complex carbohydrates are kind of where it's at. But of course, everything in moderation, including moderation. Okay, so monosaccharides. Remember I showed you glucose earlier and it looked kind of like this, where one of the corners was an oxygen and the rest were carbons and there were some hanging offy bits. That is the structural formula, so it shows you information about the true structure of glucose, which usually exists as a ring. But when you're comparing different monosaccharides side by side, like we are here, it's often useful to straighten them out so that you can compare them more easily. So that's what's going on with this. So here we have glucose and galactose, which you just got an example of, and fructose. Guess where that comes from? Fruit. So these molecules are something called isomers, and that means that they have the same chemical formula but slightly different structures. So what do I mean by chemical formula? Well, if you were to count the number of C's, H's, and O's in each of these molecules, you would find that the chemical formula for each of them is C6H12O6. So if I showed you this chemical formula, you would know that it was a monosaccharide, but not necessarily which one it was, because chemical formulas don't give you any information about the shape of a molecule. They only tell you the relative abundance of the atoms within the molecule. So not as detail rich. So if you want to link together some monosaccharides, you can make a variety of disaccharides. So sucrose, which is a common form of sugar, is a glucose plus a fructose. Lactose, which is present in dairy products, is glucose plus galactose. And maltose is glucose plus glucose. So these are disaccharides, so it's two monosaccharides holding hands. Polysaccharides are what happens when you glue together more than one monosaccharide. So for example, here we have a variety. So let's start with starch. This is a storage form of carbohydrate in plants, so usually in roots and seeds and sometimes in things called rhizomes. And humans like to eat these. So examples of rhizomes that humans consume are ginger, for example. Ginger is a rhizome. And tuberous vegetables like potatoes and turnips and rutabagas. These are also starch storage structures that plants use and that humans and other animals find palatable. So these can be formed when nutrients are in excess and then broken down into simple sugars when a plant needs to use them or when an animal eats them. Glycogen is kind of the animal cell version of starch. So it's our version. We have a storage molecule that's really similar to starch, but instead of being a straight chain of monosaccharides, it's actually a branching chain. So you have this chain here, and then it branches off to this one here. And this is actually just a subset of glycogen. If you Google image search true glycogen, it's way bigger and branchier. So this form of carbohydrate is in animals, and it's usually stored in muscle or liver cells. It's actually not really an or, but rather an and. So this can be formed for storage or broken down for use as needed. So in skeletal muscles, which are the ones you guys use and I use to do things like walk around and point, any voluntary action, they use glycogen first, which is stored near the nucleus of the cell, and then once they run out of glycogen, they switch to other fuel types. All right, sorry, I'm back. I had to pause for a moment. You probably didn't notice, but I did, because talking this long makes me very, very thirsty, and I had to take a drink, and I didn't want to subject you to the sounds of that. So let's move on. Cellulose is actually the most abundant polysaccharide on Earth, and that's because this is the structural polysaccharide in plants. So we use cellulose, I mean we humans, we use this polymer to make paper. So paper products are made out of this stuff. And when we eat cellulose, which we do when we eat certain vegetables, it's undigestible. So we don't have an enzyme that can get in here and cut this bond. No enzyme for this. In humans, anyway. Other critters, to varying extents, can do this, but not us. So even though we can't metabolize cellulose, it's still useful to eat it, an important part of diet, because it is dietary fiber, which helps keep your intestines and colon healthy and clean-ish. Chitin, or chitin if you're David Attenborough, is a support in animals like spiders, insects, and crabs. So anything with an exoskeleton, so a rigid outer skeleton as opposed to a bony inner one. And this is also indigestible to us. So next time you eat shrimp, if you are a person that eats shellfish, give the tail a few chomps. I think you'll find that it's unpalatable and hard to eat. Okay, lipids. So now we are moving on to lipids. And lipids are fats, oils, and waxes. So anything that is hydrophobic, made of carbon, hydrogen, and oxygen, and has less oxygen than carbohydrates, is going to be a lipid. And they come in various forms. So this cute little guy is a river otter. Look at that little face. So adorbs. And animals like otters and also beavers and other mammals that spend a lot of time in the water secrete hydrophobic lipids onto their fur, so that if you were to like poke your finger into here, you can see that the fur is separated into these little points here, but if you poked your finger down there, the underside would be dry. So that helps keep the animals warm by having a boundary layer of warm air when they're swimming around. Also, I can't resist. I'm going to give the otter some cute spectacles. Oh no. Cursed Microsoft line thing. All right, now he's Science Otter. Great. So, otters stay dry using science. Huzzah! So, again, note that these don't really have the same monomer-polymer format as the other ones, but there are some similarities. So, various ways that you can, for example, configure fatty acids resemble the relationship between monomers and polymers. So lipids come in several different varieties, and these include saturated and unsaturated fatty acids. So what saturated and unsaturated means is it's a way of describing whether or not within the chain of carbons, every single carbon is bound to the maximum number of hydrogens. So this is saturated because each carbon is bound to at least one other carbon, typically two, unless it's the end two, and the other available bonds are taken up by hydrogen. If two of the carbons choose to hold hands twice, so they're doubly covalently bound to each other, that means if we count bonds, 1, 2, 3, 4. So all of the valence needs of this carbon are met, but they are not being all met by hydrogen. They are being met by two carbons bonding to each other twice. So because of this, they are not bound to two hydrogens each, only one, meaning that there is not the maximum amount of hydrogen in that fatty acid. So they're unsaturated because they're unsaturated with hydrogen. And in this case, they're saturated because they're saturated with hydrogen. So, like I said, you can use saturated and unsaturated fatty acids to build other molecules that are similar but not identical. So the first kind of these is triglycerides. And before we move on to that, it's important to look at the components of fatty acids. So these are long chains of hydrocarbons, usually between 12 and 18 carbons. And the reason it's an acid is the carboxyl group. So this is something I forgot to mention. The carboxyl group is a name for this configuration. So when we write the chemical formula for that, we just write this which tells you what's in there. It doesn't tell you the structural arrangement of it. But the reason these are acids is because this hydrogen dissociates in solution to form H+. So these both give off protons in solution. So this is an important component of triglycerides and of phospholipids. So let's start with triglycerides. So the tri comes from the fact that there are 1, 2, 3 fatty acids in a triglyceride, and they are bound to a glycerol, which is a 3-carbon molecule related to glycerin. So glycerin is the major component of a lot of soaps and body washes, and glycerol is similar in nature to that. So triglycerides are the storage form of fat in all cells. And I mean all here. So in animals, adipocytes; in plants, seeds. So these can be formed when nutrients are in excess and broken down for use when needed. And because both carbohydrates and fats are hydrocarbon in nature, you can actually turn sugar into fat. So if you eat too much dietary sugar, it will get converted into triglycerides and then stored in your adipocytes. So it's not the case that you can eat as much sugar as you want and it'll all get converted to glycogen, lucky you. Instead, if you overeat sugar, your body will sense that you are in a period of a special food wealth, and it will turn that sugar into fat instead. So there are different kinds of triglycerides, and they have different fatty acid chains, but there's always this glycerol head. So there's 2 types of fatty acid chains, as we discussed, saturated and unsaturated. And depending on what kinds of fatty acids are in the triglycerides can either be saturated or unsaturated as well. So for example, and let's erase all my markups here so you can look, this is a monounsaturated triglyceride with a monounsaturated fatty acid. So the reason I'm using the word mono is because a monounsaturated fatty acid is one in which there is only one pair of carbons doubly bound to each other. A monounsaturated triglyceride is therefore a triglyceride in which there is one unsaturated fatty acid and 2 saturated ones. All right, so this is just a restatement of the stuff I said earlier. Remember that saturated fatty acids are saturated with hydrogens, so they are straight. And because of this property, they can pack very tightly into a triglyceride, so they're denser. So if I were to line a bunch of saturated fatty acids up next to each other, I can nest them really closely because they're all the same shape. So because that permits tight stacking, they're denser and they're usually solid at room temperature. So examples of saturated fats that we use and eat in our diet, those include things like lard, Crisco, which I find to be offensive, but it's useful for baking, I suppose. I just think it's gross. And butter or margarine. So you shouldn't really eat these in large amounts because they tend to form plaques on the insides of your arteries and cause heart disease. But like I said, everything in moderation, including moderation. So that one piece of cheesecake or cheese plate or foie gras, if you swing that way as far as food tastes, that's not going to kill you. You just need to make sure that you moderate your saturated fat intake. Unsaturated fatty acids are ones with one or more doubly bound set of carbons. So monounsaturated has 1, so this would be a monounsaturated fatty acid. So let's add mono to that. And polyunsaturated ones have more than 1 double bond. So because of this little kink in the chain, there's an angle here. Angle. And that means you can't stack them as densely in the same way. So if I tried to stack another one on top of this one, I'd end up with more space between them because they are not as able to pack tightly. And that's why they are liquid at room temperature. So olive oil and other cooking oils are unsaturated, and these are a healthier dietary option. Phospholipids are very important. These are consisting of one glycerol, so familiar. 3-carbon molecule, 2 fatty acids, also familiar, and in many phospholipids, there is 1 saturated and 1 unsaturated fatty acid. Now we're getting into some new territory here. So before, or up until now rather, we've sort of characterized molecules as being either polar and therefore hydrophilic, or nonpolar and therefore hydrophobic. That's not quite accurate for all molecules. So there's a kind of molecule that is called amphipathic. And that means that it has components that are hydrophobic and components that are hydrophilic. So the reason that this has a hydrophilic area is because phosphate, PO4, is an ion, so it's PO4 minus. And that means that there is a plus here and a minus here, so the head is hydrophilic. These tails, however, are not hydrophilic. They are hydrophobic. So when a bilayer is formed, the hydrophobic tails face each other, and the hydrophilic heads face either inside of the cell or outside of the cell. And this creates the opportunity to form a little bag of water, which is what a cell is. It also helps to determine what kinds of molecules can pass through the cell membranes. So we'll get to that later when we talk about membrane transport. But let's look at some phospholipids in a bilayer. So bi means 2. That means that there is not 1 but 2 layers, or a sandwich, of phospholipids. So the hydrophilic heads are out here, and the hydrophobic tails are afraid of the mean, scary water. Let's make the water look mean. And so the hydrophobic tails hide in this middle layer. So it's kind of like a sandwich. Okay, steroids. So steroids are also important, and that's because we use these to make hormones. So hormones like estrogen and testosterone, and a bunch of other hormones as well. So if you take A&P, you'll learn all about those, probably more than you want to know, so stay tuned for that. So these are actually arranged into rings rather than chains. And specifically, steroid lipids always have 1, 2, 3, 4 rings. And if you were to count the carbons, there would be 17 of them. So that's kind of the rule for steroids. The parent steroid is cholesterol, which is what this is. So cholesterol is what we use, our bodies use, to make hormones. And it's also used to synthesize important vitamins and stabilize cell membranes. So although cholesterol dietarily has gotten sort of a bad rap, it's actually a really useful molecule as long as you don't overdo it. And finally, waxes. So these are long hydrocarbons, so more than 12 to 18 carbon chains, and they have a hydroxide group and a fatty acid. And these are used for waterproofing and protection. So beeswax is a kind of wax. Excuse me. So is lanolin, which comes from wool. And lanolin is actually pretty similar in its components, wax-wise, to the waxy substances that our sebaceous glands, human sebaceous glands, secrete, because we're mammals and so are sheep. And so you may see lanolin sold in stores as a moisturizer. So you're basically taking sheep wax and waxing yourself with it. Humans make earwax, and that is a waxy, bitter substance that helps keep insects from hanging out in our nice, dark, warm ears. I know, gross, right? And carnauba wax, which comes from a plant and is typically an ingredient in car wax. So it helps protect your paint by putting a boundary layer of wax between your car paint and the elements. So now let's talk about proteins. And I want to stress, and I can't stress enough really, how important proteins are. So I mentioned enzymes earlier. Every chemical reaction that is critical for our survival as animals, both within and between our cells, is carried out by enzymes, which are a kind of protein that makes chemical reactions more likely to happen. So no enzymes, no life. And all cells contain thousands of different kinds of proteins, each with their own unique structure and function. So here are some protein sources. Here we have some salmon, some eggs, some beef, some chicken, I assume that's chicken, and legumes and nuts. So those are some sources of protein. Proteins are made from amino acids. So you actually have already seen an amino acid in the beginning when I was describing hydrocarbons and different kinds of them, but they have a fundamental structure. So amino acids have a central carbon, which is this one right here. And those are bonded to an amino group, which is a nitrogen bound to two hydrogens. And I know it says H2N here. What it really looks like if you were to draw it out is this. The C is bound to the N, and the N is bound to two hydrogens, like that. And a carboxyl group, so there's another C at the end of this, and that C is double bound to one oxygen and bound to an OH group here. Then there's a hydrogen and an R group. So I want to stress that the R doesn't stand for any element in particular or an atom. It's a placeholder for can be anything. So the portion of an amino acid that remains consistent across all amino acids is this part. But what the R group looks like depends on what amino acid it is. So here, let's look at some examples. So one of the simplest amino acids is alanine, so it just has a methyl group down here. Valine is a little bit more complicated, so it has 1, 2, 3 methyl groups. So methyl equals CH3. And these don't really exist by themselves because, of course, carbon would want to form that fourth bond and turn it into methane. So methyl is typically a group of a carbon and 3 hydrogens that hangs off the end of something. Lysine has a chain with 4 repetitions of CH2 ending in another amino group. And aspartic acid has 1 iteration of CH2 and another carboxyl group. So aspartic acid is twice as acidic as regular amino acids. And lysine has more nitrogen than other amino acids. So the properties of the amino acids really depend on their R groups. And that's what this box says as well. I just think I said it fancier and better, in my opinion. Okay, so let's say you want to build a protein. Well, you have to glue a bunch of amino acids together, and we call this a polypeptide chain. So remember, peptide is the kind of bond, not the name for an amino acid. So when you make a chain of these, the next step is to fold up the chain. I'm pointing this out here because cysteine is kind of a special case. Cysteine has sulfur in its side chain, and sulfur likes to bond covalently with other sulfur. So that forms a really stable, covalently bound bridge called a disulfide bridge. Okay, so the next step after you build a rope is to fold your peptide chain up. So I showed you a disulfide bridge, but they're not the only kind of interaction that facilitates protein folding. And in lecture, I gave you the hammer analogy, and that was just an elaborate way of proving to you that the shape of things determines their function. So a hammer is shaped perfectly for doing nail-related tasks like pounding nails in and taking nails out, but it's really bad at other stuff that it's not specifically designed for. And proteins are the same way. So a protein that is just a rope of amino acids, not useful. You have to fold it up and then fold the folds and then fold the folded folds to get a useful protein. So mature, functional protein. Voila. Okay, so let's go over the structures. So protein structure gets increasingly complex, and we just name these according to their level of complexity. So primary, secondary, tertiary, and quaternary. So the amino acid order is the primary protein structure, and this is determined by DNA. The peptide backbone of the chain of amino acids, because remember, technically this is a polypeptide, the peptide backbone will result in hydrogen bonds attracting different areas of the chain, and that causes the chain to fold up into a particular configuration, either into a spiral, which we call alpha helices, or a beta sheet, where maybe this is hydrogen bound to that, and that is hydrogen bound to that, etc. So it corrugates the sheet instead of spiraling it. Regardless, tertiary protein is when you take these helices and sheets formed by the folding of the amino acid chain, and then you fold those up. So this region consists of an alpha helix, and so does this one, whereas this is an area with a beta sheet. And that gives you a globular folded up protein. So we're almost done. Most large proteins are actually composites of more than one amino acid chain. So you need multiple what we call subunits that are similar to each other to come together and interact to form quaternary protein structure. So this is when there are more than one folded amino acid chains bound together. So because structure equals function, that means that if you undo structure, you ablate function. So if you lose secondary, tertiary, or quaternary structure, this is called denaturation, which doesn't mean breaking peptide bonds. It just means unfolding. So to denature something is to take a protein from this state and turn it back into this state. So unfold the guy. And this makes its function go away. So denaturation typically happens due to changes in temperature, pH, or due to the action of enzymes. So an example of a temperature change denaturation reaction that everybody's probably observed is cooking eggs. So when you cook an egg, the albumin, which is a protein, in the egg white denatures and then cross-links with other denatured albumins, and that's why it goes from clear to white. Okay, home stretch. I'm sure you're tired of watching, and I am growing weary of talking, so this is the last little chunk of the biological macromolecules lecture. So here we have a double helical structure, and this is a portion of a DNA molecule. So we call this the DNA double helix. And it's a double helix because it looks like a twisted up ladder. So a single helix is more like an alpha helix. That would just be one of these strands in a spiral, but the double helix is this configuration. So the people that discovered the nucleotide bases and the rules for their pairing were Watson and Crick. So they're very famous for having discovered and elucidated the nucleotides and base pairing rules. The person who helped figure out that it was a double helix and therefore a spiral was Rosalind Franklin. And Rosalind used X-ray crystallography, which is an imaging technique, to take pictures of the DNA molecule in various poses, so to speak, so from various angles. And in a couple of those images, what she saw was an X, which is interesting. So that picture was taken from this angle, so looking at the molecule sort of down the barrel of it, and if you can pivot this in your mind, you'll be able to think, oh yeah, so that would be an X. So she figured that out. All right, so nucleic acids are made from monomer nucleotides, and a nucleotide is a complex monomer with 3 repeating parts. So there is a phosphate group, which is PO4 minus or 2 minus. And this phosphate group is giving the whole molecule polarity. So because these O's are charged, that means that this is polar. The second part is a pentose sugar. So pent means 5, and if we count the sides, there are 1, 2, 3, 4, 5. So this is not like a hexose sugar like glucose. This is instead a deoxyribose or ribose. And the last part is a nitrogenous base, and these vary. So it's called a nitrogenous base because it's a double or single ring structure where some of the ring members are nitrogens. And there are particular rules for these, which I'll show you in a minute. So the DNA double helix, if you were to straighten out that helix and make it more of a ladder, you'll see that there is a repeating sugar phosphate backbone. So it goes phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, all the way down. And the sugar phosphate backbone is by definition water soluble because it is charged. The nitrogenous bases are in the middle of the ladder, and these are the parts that interact with each other to bind one strand of DNA to another. So DNA is a double-stranded helix. This is one strand, and this is the second strand. So the nitrogenous bases include thymine, cytosine, adenine, and guanine. So C, T, G, and A. And there are specific pairing rules for these bases. So A always goes with T. And G always goes with C. Now, it doesn't really matter which side is which, so you can see that these two pairs have the same members, but they're flipped, and same with these two. But you'll never see a G with a T or a G with an A, and you'll never see a C with an A or a C with a G. Also notice these dotted lines here. So these dotted lines represent hydrogen bonds between the nitrogenous bases. Between T and A, between T and A, there are 2 hydrogen bonds. Between C and G, there are 3. So that means that it's a little bit harder to pull C and G apart from each other than it is to pull T and A apart from each other. Now, why would you want to pull the strands apart at all? Well, we'll get to that a little bit later, but the Cliff's Notes version, the short version, is that if you want to make a protein, the information is stored in the order of nucleotides. And the way to access that nucleotide order is to unzip strand A from strand B. So what that means is you have to have an easy way to get the strands apart, but a stable way for them to stick together. And because hydrogen bonds are individually weak, but collectively and cumulatively very, very strong, having DNA bound together at the center with hydrogen bonds is a good way to make it easy to unzip when needed, but stable in its configuration when it's not. So DNA versus RNA we'll get to in more detail when we start to talk about how proteins are made and how the genome is used to form phenotypes, which are physical characteristics. But until then, let's just make some basic distinctions between DNA and RNA. So DNA is permanent, meaning that it stays put. It stays in the nucleus specifically. So the nucleus is a central membrane-enclosed location in a cell, and it's double-stranded, as we saw. So this is the genetic code. So the Human Genome Project mapped every gene in the human genome. And in every cell, for the most part, DNA stays in the nucleus and stays there all the time. RNA is a little bit different. So this is temporary, and it's temporary because RNA is made using DNA templates, and then it's used to make proteins after that. So we'll talk about how that happens in some subsequent chapters and lectures, but for now we're just making a distinction. So it's temporary. It's used to turn the genetic code into protein, so this is part of the protein production or making machinery. And it's located both in the nucleus and the cytoplasm. So it's made in the nucleus and then it goes into the cytoplasm to do its job. And it's single-stranded. Another difference is the D versus the R. So deoxyribose is a kind of ribose with 1 oxygen missing. Ribose is just regular ribose, so it has the oxygen that deoxyribose lacks. So that's the difference there. So, just to review, biological macromolecules are diverse, and they have different structures and functions, but they also have a lot in common. And they each play very specific and very irreplaceable roles in biology of cells. So as we move on to contents of cells and structure of cells, we will include biological macromolecules in all of our discussions about those. And that is the end of the slideshow. So thank you for your attention and happy studying for the upcoming midterm.